Hello students, I am teacher Svetlana and in our today's lecture we will learn about improvement in food production, biofortification and microbes in human welfare, which is covered in the chapter Enhancement of Food Production. So let's begin. Improvement in food production. Students, food is one of the basic needs as it gives us energy for everything. It keeps us alive, strong and healthy. It can be defined as anything solid or liquid which when swallowed, digested and assimilated in the body, it helps in keeping us well. It is organic, energy-rich, non-poisonous, edible and nourishing substance. Green plants synthesize their own food through photosynthesis, but animals, including humans, cannot synthesize their food on their own, hence are dependent on plants directly. Now, students, we have situations like the famine, which is responsible for dearth of food, besides the rapid and dramatic increase in the world population over time. So, we have natural calamities which affect our food production, and at the same time there is a huge population which has to be fed. So to meet the increasing demand for food, there is need for improvement of food production, both quantity wise and quality wise. That is the nutritive benefits that we draw from our food. So there is two important ways which are explored to increase the food production, that is plant breeding and animal breedings. You can see here in this image. So most of the crops and livestock today are produced by selective breeding. Biofortification. Students, it is a method in which crops are bred, that is, they are produced for having higher levels of vitamins, minerals, and fats. That is, we improvise their nutritive value. Due to this, problem of malnutrition can be overcome. Following objectives were considered for the breeding program: protein content and quality, oil content and quality, vitamin content, micronutrient content, and the overall quality. Biofortification can be achieved through conventional selective breeding practices and also through recombinant DNA technology. It focuses on making plant food more nutritive as the plant grows or develops. Have a look at this image here. It shows you what is normal fortified food is and what is a biofortified food product. Then in normal fortification, you can see that the grain is harvested, then it is processed by adding the required nutrients to it. So nutrients are added during processing, but you can see in the biofortified plants, you can see the nutrients are introduced into the plant when it is growing. So then the harvest that you get is rich in all the essential nutrients. So this process is called as biofortification. Now some examples of biofortifications: fortified maize having twice the amount of amino acid lysine and tryptophan. Wheat, which is named as Atlas 66, has a high protein content, and iron fortified rice has 5 times more ions are developed. Vegetable crops like carrot and spinach having more vitamin A and minerals are developed. Vitamin C enriched bitter gourd tomatoes have been developed by the IARI. So this process is called as biofortification. Microbes in human welfare. Biotechnology is the application of scientific and engineering principles for the processing of materials by biological agents to provide goods and services to humans or for human welfare. So to put it in a nutshell, students, in biotechnology, we use microorganisms or any of the microbial products for the use of human purpose or for human welfare. Now, there are a variety of microorganisms like algae, fungi, bacteria, viruses, protozoans, nematodes, etc. And they are products that exhibit beneficial activities which are used for welfare of humans in regard to food, health, industry, agriculture, medicines, biocontrols, etc. These organisms are also used variously in food and feed technology, industry, waste utilization, etc. And also they are used for the purpose of energy. So here we are going to explore what are these microbes and their uses in food, sewage treatment, energy in this chapter. Microbes in food preparation. The development of biotechnology occurred in two phases, which are traditional till 1971 and the modern after 1970. Traditional biotechnology is based on the fermentation principle by using fermenting bacteria. They were used in the preparation of variety of indigenous fermented food products. Have a look at this image here. It shows you the fermentations process of fermentation, where it highlights the different organisms that are used and the products, the fermented products that we obtain from them. That is, here is the Swiss cheese and the bacterium, that is Propionibacterium, which is used in Swiss cheese. Then we have Aspergillus, Lactobacillus, Streptococcus, which give lactic acid, which are used for the production of cheddar cheese, yogurt, soy sauce. Then you have Saccharomyces, which give you carbon dioxide and ethanol, which are used in preparations of beverages like wine, beer. Then you have Clostridium, which gives you acetone, isopropylone, which is used for the preparations of nail polish removers, rubbing alcohol. Then you have Escherichia acetobacter, which gives us acetic acid, which is used in the production of vinegar. So a number of these fermented products are formed because of these involvement of these microorganisms. So here we talk about the use of microbes in food preparations. Here, students, we will have a look at the different food preparations which involve use of microbes. The most common and Indian food delicacies, which is the dosa, dokla, and idli. Now, these are fermented products produced due to the activities of bacteria. They are fermented preparations of rice and black gram with airborne leuconostoc and streptococcus species of bacteria. Carbon dioxide is produced during fermentation, causing puffing up of the dough. Then we have microbes as a source of food. Now, some microbes are and their fruiting bodies are directly used as source of food as they are rich in vitamins and proteins. They are termed as SCP, which is also called as single cell proteins. Here, have a look at this image. It shows you the single cell proteins. Now, the term SCP or the single cell protein denotes dead and dried cells of microbes like bacteria, algae, molds, and yeasts. We also have some mushrooms and truffles which are directly used as food. They belong to the higher fungi. They produce large, fleshy, fruity bodies which are edible. Fruiting bodies are sugar-free, fat-free, but rich in proteins, vitamins, minerals, and amino acids. The food in the fruiting body is low-caloried. Then we have the third important category where microbes are used in food preparation, that is the dairy products. So lactic acid bacteria, which are termed as LAB, like lactobacilli, are added to milk. It ferments lactose sugar of milk into lactic acid. Lactic acid thus causes coagulation and partial digestion of the milk protein, that is casein. Milk is changed into curd, yogurt, and cheese. The starter or the inoculum used in preparation of milk produces actually contains millions of lactic acid bacteria. So when you have the start or the inoculum used in the preparation of milk products actually contains a lot of lactic acid bacteria. If you must have seen your moms preparing curd at home, you will see that to milk to warm milk, she will add some amount of the leftover curd, which acts as an inoculum. Now, that inoculum has a large number of lactic acid bacteria. So, let us see how curd is prepared in India. Curd is prepared by inoculating milk with Lactobacillus acidophilus. It also checks growth of disease-causing microbes. We have yogurt. Yogurt is produced by curdling milk with the help of the organisms called Streptococcus thermophilus and Lactobacillus bulgaricus. Then we have buttermilk. The acidulated liquid left over after churning butter from curd is called as buttermilk. And last but not the least, we have cheese. The milk is coagulated with lactic acid bacteria and the curd formed is filtered to separate the whey. The solid mass is then ripened with growth of mold that develops the flavor in it. Different varieties of cheese are known for their characteristic texture, flavor, and taste, which are developed by different species of microbes. Students, the Roquefort and the Camembert cheese are ripened by blue-green molds. Here, have a look at this cheese here in this image. They are ripened by certain blue-green molds called as Penicillium roqueforti and Penicillium camemberti respectively. And if you observe this cheese here, which is called as the Swiss cheese, if you closely see, you can observe large holes in this cheese. These are developed due to the production of large amounts of carbon dioxide by a bacterium known as Propionibacterium shermani. So this is how microbes have had a role to play in the production of different kind of food and large amounts of food industries, especially the dairy industry, is dependent on the use of microbes as one of their key ingredients in the preparations of the different types of products that they deliver. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.